Hello, I am Jongho Moon from the Department of Physics and IBS CMSD Research Center at Korea University. In this video, I would like to introduce a ultra-fast imaging system that can see phenomena occurring at the speed of light. We are interested in the motion of fast-moving objects. The trajectory of a basketball like this can be taken even on our mobile phones. You just need to have a shooting speed of about 30 frames. These pictures of bullets passing through an apple require an imaging system that can capture at hundreds of thousands of frames per second. So how do we see an object moving at the speed of light? This is a famous image of light passing through a Coca-Cola bottle from MIT Media Lab. To obtain this image, they need an imaging system with an imaging speed of several trillion hertz. However, in the case of this image, it would have taken less than 1 nanosecond for light to pass through that Coca-Cola bottle, but it is possible to reconstruct the data obtained by passing the light through the Coca-Cola bottle hundreds of times. It's the fastest, but slowest picture in the world. In this video, I want to talk about an imaging system that can acquire such a light trajectory in a single shot. Rather than explaining the concept of our high-speed camera system in time, I would like to compare it to a color imaging system. In fact, each of the RGB colors is a signal that is several hundred femtoseconds apart. We have a color object composed of these letters IBS. How do we color imaging with a monochrome, that is, a color indistinguishable camera? First of all, there may be a way to do this: multiple acquisitions with different lighting colors and filters. You can use multiple cameras to distinguish colors with different filters. Or you can split the pixels in single camera and separate the colors by receiving different colors of light in different parts. Also, although it is difficult to explain in detail here, it is also possible to acquire color images at once through a compressed sensing method through an optical system and computer processing. However, you may lose some color resolution. These various color imaging methods just mentioned are intended to easily talk about color imaging as a representative method used in ultra-fast camera research that can actually see the trajectory of light. For color, a color camera can be used, but since there is no 2D detector that is fast enough to image the trajectory of light, these methods are used. Since the fastest 2D camera that exists can only take 1 million pictures per second, many people are researching a ultra-fast camera technique through various optical systems and algorithms. There are pump probe imaging methods that measure multiple times, stamp imaging by splitting light and image sensors, and cup using compressed sensing technology. Pump probe has the disadvantage that it is not a single-shot method, and stamp and cup are excellent imaging systems that can obtain high-speed images at once, but have the disadvantage of sacrificing spatio-temporal resolution. Our spatial frequency division multiplexing technique, which we are going to introduce now, is a way to get high-speed camera images in a single shot without loss of spatio-temporal resolution. Using a low-coherence laser in an interferometric holography system such as OCT allows you to only see a signal of light from a time exactly matching the reference. Based on such an interferometer system, even if there are detectors that cannot distinguish different colors from the IBS object and signals from different times, if you put the coherent color and time reference beam at different angles like this, in the camera raw image, they are overlapped and indistinguishable, but in the spatial frequency domain through the Fourier transform, information of different colors and time appears separated in different spatial frequency bands. If you do inverse Fourier transform these one by one, you can distinguish different color and time information from a single camera image. This is a simple experimental setup drawing. It uses a single pulse from a strong Ti-sapphire laser. If you look at the reference first, the beams at different angles are created through the 2D grating that is floated on the spatial light modulator, and the glass echelon with different thickness goes through different delays and enters the camera at different times. It made a lot of laser beams at different angles and different times. The sample line that has the actual imaging information is again divided into a pump and probe arm. In the pump arm, it is used to create ultrafast phenomena. The probe arm is actually imaging. As such, a signal with the ultrafast image information enters the camera and interferes with the reference beam. As shown separately, the reference beam and sample beam enter the camera at exactly the same time. The propagation of light was imaged using the system described so far. In this way, only one line of trajectory is visible in the camera raw image, but information of different times is separated in the spatial frequency domain. Therefore, through an inverse Fourier transform, light trajectory can be viewed at 200 femtosecond intervals, at a frame rate of 5 trillion frames per second. We could also see the trajectory of the light being attenuated as it propagated through the complex medium.